Hello, welcome to a webinar series on precision imaging agents and diagnostics. My name is Ilman. I'm an assistant professor of radiology and radiological science at Johns Hopkins University. I am delighted to present this webinar series. My talk is going to be on imaging therapeutic immune cells today. Let me start off with introducing the concept of cancer immunoediting. When cancer develops in our body, our immune system tries to fight against the cancer to eliminate them using a variety of our immune components. Cancer continues to fight against our immune system where they can reach equilibrium, and eventually when they won the battle, they escape from our immune system and became tumor. Cancer immunotherapy is an effort to revert this process back down to elimination using components of our own immune system. To introduce many different approaches of cancer immunotherapy, I show this nice cartoon where it dissects cancer immunity cycle into 7 different steps, starting from release of cancer cell antigen presentation to the immune system, and priming activation of T cells at the lymph node, trafficking of those active T cells to tumor, infiltrating T cells into the tumor through the blood vessel, and recognition of cancer cells by T cells and killing of them. The field of cancer immunotherapy has developed a variety of different approaches attacking and utilizing each and every step. For example, majority of cancer therapy that can cause immunologic cell death or part of the cancer immunotherapy. To activate cancer antigen presentation, concept of cancer vaccines and recombinant cytokines has been used. For priming activation, immune modulatory antibodies and recombinant cytokine has been tested. To boost trafficking and infiltration, chemokines and anti-VEGF receptor treatment has been tried. And many different types of T cells, engineered CAR Ts, NKs, tumor-infiltrating lymphocyte, and many others has been vigorously tried. And lastly, to enhance cancer cell killing, checkpoint inhibitors and anti-myeloid-derived suppressed cells therapy has been tested. Despite the success of these cancer immunotherapy in certain types of cancer, there are still many hurdles to overcome. For example, checkpoint blockade therapy, there are variable responses among different cancer types, and there's a population of non-responders. For cell-based therapies, majority of solid cancer efficacy has been poor, and there are severe toxicity accompanied with cell-based therapies. Molecular imaging and immunotherapy can provide critical information for these therapies to improve their efficacy and safety. I'll give you some examples on how molecular imaging can help improving these cancer immunotherapies. First, for example, for checkpoint inhibitors, if one can predict the therapy responses before the treatment, it'll be very helpful. And this study analyzed which factor can determine the efficacy of anti-PD-1 and PD-L1 therapy. The study analyzed 36 different variables in 3 distinct classes, which includes tumor neoantigens, tumor microenvironment and inflammation, and checkpoint targets. What they found is that the status of CD8 plus T cell infiltration into the tumor can determine the efficacy of this therapy. The more T cells are there, the treatment is going to work better. So if we could determine the status of T cell infiltration for the treatment of this therapy, it'll be very helpful to predict or even select the target patients. So these are the list of T cell targeting PET probes that are been in clinic or preclinical stage to non-invasively image and quantify and localize T cells using the PET imaging. For cell-based therapy, molecular imaging can also provide multiple variable informations. For example, we can localize where they are and quantify and determine the functional status of infused therapeutic cells. Also, we can use this information to rapidly determine therapeutic efficacy after the infusion of the cells. These imaging techniques will have to be very sensitive and selective, and engineering we do should not affect the original function of the cells. Also, it will have to be applicable to humans. In addition, our engineering, if it can be equipped with cell killing mechanism when the infused cells cause severe toxicity, it'll be very helpful as well. There are two different ways to image these therapeutic immune cells. The first one is direct labeling, where you label the cells before infusion. This is an example of preclinical study using zirconium-89 labeled auxin for in vitro tracking of CAR T cells. As you can see on the panel B, you can see labeled T cells that are infiltrating into the tumors, and then in vivo imaging in A here in the green arrow, you can detect T cells that are infiltrated into the tumor using PET imaging. The second method to image therapeutic immune cell is reporter gene-based imaging or molecular genetic imaging, where we engineer the therapeutic cells to express reporter gene before we infuse these cells, and then we image them using the probe that respond to our reporter. Advantage of this method, we can do longitudinal imaging, and we can do live cell imaging because only live cell will express this reporter gene. And based on the selection of the reporter, we can do multifunctional imaging. The drawback of this method is manufacturing can be complicated depending on the method of engineering. The reporter can be silenced. If we don't use human protein as a reporter, it can cause immunogenicity after infusion. And also there is a chance for developing tumor of these infused cells. This table introduces 4 major categories of reporters that has been developed by the field of molecular imaging. They are enzymes, transmembrane receptors, transmembrane transporters, and optical imaging proteins. Each reporter also has a specific set of its probe that are specific to its reporter, and then these have been used and tested for multiple imaging modalities. Now I'll give you some examples of reporters being used to image therapeutic immune cells. This one, the cytotoxic T cells has been engineered to express its therapeutic component in addition to reporter protein, which is HSV1-TK, and used to image glioblastoma patient treatment. If you look at pre and post CTL, you can see enhanced signal from HSV-TK, which indicates T cell infiltration into the brain. And this is another example of different patient. Also, you can see enhanced signal, which indicates infiltration of the cytotoxic T cells. The second reporter example is transmembrane receptor. In this preclinical animal model, they built CAR T cells to express somatostatin receptor 2 as a reporter and used DOTATOC as a PET probe. As you can see here in the left panel, where the tumor is, there are PET signal which indicates T cell infiltration, CAR T cell infiltration. And on the right panel also shows the imaging of T cells in tumor. The detection sensitivity of this particular model is 50,000 cells in 100 microliter volume. The third example of the reporter is transporter, which is human sodium iodide importer, where they use technetium-99 pertechnetate as a probe. In their in vivo phantom study, they show that they can detect 15,000 cells in 100 microliter volume. In their preclinical animal model, they were able to detect T cells co-localized to the tumor on day 21 post-injection of the T cells. Another preclinical example of using human sodium iodide importer as a reporter. In this case, they use a 15 labeled tetrafluoroborate as a probe, and they determined in vitro phantom study detection sensitivity of 3,000 cells per pellet. Also, in their in vivo model, they were able to detect T cells infiltrating into the tumor, and compared to the higher uptake compared to a non-T cell injected model. Recently, Mark Selmay's lab at UPenn published a new reporter, which is bacterial enzyme dehydrofolate reductase, using F18 labeled trimethoprim as a PET probe, and particularly they were able to engineer multiple reporters so that they can do multimodal imaging. They were able to detect the CAR T cell targeting GD2 in the preclinical animal model, and their detection sensitivity was about 11,000 cells per 1 microliter volume. I'd like to take this opportunity to introduce our own work for developing engineered CAR T cells for in vivo tracking. Why do you want to do this? After infusion of a CAR T cell, particularly in patient, information such as are they alive and where do they home to after the infusion? If they are there, are they functional? How long do they persist? This kind of information cannot easily obtained. And particularly, the current clinical practice to quantify infused CAR T cell is take the peripheral blood and do the analysis of CAR T cell in the blood. Would this method valid? For this, we chose prostate-specific membrane antigen as our choice of reporter for the following reasons. PSMA is a well-established prostate cancer marker, and a high-specific PET probe, which is F18 labeled DCF-PYL, has been approved by US FDA. And we also have developed many therapeutic probe that can target PSMA using multiple variety of radionuclide. And also, we developed a pro-drug that can target specifically PSMA expressing cells. When we consider PSMA as a choice of a reporter, there is one concern, which is PSMA being type 2 transmembrane domain, and there is a motif from amino acid position 1 to 5 that can trigger the internalization. Also, there has been reports that putative signaling via MAP kinase and PI3 kinase AKT pathway may trigger the enhanced proliferation and growth survival of PSMA expressing cells. So when we introduce this wild-type PSMA into T cell, there is a chance that this PSMA may make the T cells into tumors. So we developed a strategy to prevent that, which are introducing one amino acid mutation at the N-terminal terminus, or deleting 9 amino acid from the N-terminus to create a truncated form of the PSMA. So we created a CAR T cell that targets CD19 blood tumor, expressing blood tumor, by introducing this construct, which express a CAR construct and the PSMA. We made 3 different constructs for wild type and 1 point mutation and 9 amino-deleted version. We confirmed that these T cells express high level of PSMA on the surface by using FACS analysis. After completion of the engineering, T cells expressing 3 different types of PSMA, we test if expression of PSMA may affect the function of cytotoxic T cells in vitro, and confirm that there's no hindrance of cytotoxicity for expression of all 3 types of PSMA. And the cytotoxic effect is proportional to the amount of effector, which confirms the cytotoxicity comes from the CAR construct we engineered. To test this engineered CAR T cell in vivo model of CD19 expressing tumor, we developed this model by injecting NAM6, which express GFP and 5-fluoroluciferase as a dual reporter, subcutaneously into immunocompromised mice. When we inject them, it develops spontaneous metastable lesions in spleen, liver, and bone, which were shown in this bioluminescent imaging. And this model provides targets for CAR T cells for both local and metastatic tumors. Since we didn't see any difference in terms of in vitro efficacy from T cells with 3 different PSMA constructs, we chose to use truncated PSMA, which has 9 amino acid deletion for our further study. We went on and tested efficacy in vivo using this model and compared the efficacy with non-treated and mice treated with mock T cells. And all of mice that treated with our CD19 truncated PSMA CAR T cell were cured compared to the other 2 groups that were lethal because of the injected tumors. Very encouraged by our in vivo efficacy study, we went on and evaluated the sensitivity of detection of our engineered CAR T cells. In vitro phantom studies show that we were able to detect about 2,000 cells in 20 microgram volume using preclinical PET scanner. Also, when we implanted different number of T cells in shoulder of mouse, we were able to detect about 2,000 cells in 15 microliter volume using preclinical PET scanner, which demonstrate a superior detection sensitivity compared to other PET imaging studies of CAR T cells. Next, we performed longitudinal PET-CT study in our preclinical model, where we try to recapitulate in a clinical scenario. You are seeing 2 different imaging modalities. One, bioluminescent imaging, which demonstrates tumor burden, signals coming from the tumor cells, and in the middle of each panel, PET-CT images and PET signals coming from the T cells by PSMA and POIL combination. On each panel on the left, day 0 is a tumor burden before we treat the mice with the CAR T cells. And in the middle, we show a PET-CT imaging at indicated date, and then on the right, you see bioluminescent imaging 1 day post of our PET-CT imaging. So at day 4, post-injection of our CAR T cells, our CAR T cells start to accumulate at the bone marrow here in the hind limb, where the tumors are metastasized to. And at day 5, you see the cleared BLI signal from the mice, which indicates the elimination of tumor bar CAR T cells. And it continues to happen day 5 and day 6, and there are increased number of CAR T cells at the bone marrow and complete remission of tumors from the bone marrow. At day 7, post-injection of CAR T cell, after removing all the bone marrow tumors, CAR T cell starts to move and accumulate at the site of tumor here at the local tumor, and you see obvious accumulation of CAR T cell at day 8 and decrease of tumors from no site. And day 9 and day 10, there are no tumors from the injection site, which indicate CAR T cells all moved there and successfully eliminate all the tumors from the mouse. And day 11, day 12, the mouse were cleared from the tumor, which indicates the efficacy of this CAR T therapy. And this longitudinal study shows location and movement and quantification of infused CAR T cell in our animal model. And as I mentioned earlier, the current clinical practice to quantify infused T cells is to take patient peripheral blood and count how many CAR T cells are in the blood. The question was if that method valid, which means does that really show what's going on in the the patient infused with the CAR T cells. So we designed a study where we try to correlate the amount of T cells in the peripheral blood, also bone marrow, and correlate them with actual number of T cells, which were determined by our PET imaging. As you see here in this data, there is no correlation between number of peripheral blood CAR T cells and the number of T cells infiltrated into the local tumor. Also, those for the bone marrow. And this indicate that just taking patient peripheral blood and quantify how many cells in the blood does not necessarily recapitulate what's really happening in the body of infused patient. And also that indicate that our PET imaging, which can provide quantitative information, may be more precise way to determine pharmacokinetics of infused CAR T cell in the patient. In this presentation, I introduced method to imaging therapeutic immune cells. There are two different methods which has unique use for them. For direct labeling, this method will be suitable for imaging non-engineered cells in human patients. For reporter-based imaging, there are more things to do, but this method will have to be carefully designed and executed in that the reporter may have to be may have to have minimal expression in normal tissue, and it should not be immunogenic, and the detection sensitivity and specificity should be higher, and engineering reporter gene should not interfere with the cytotoxic function of the infused cells, and it'd be better suited for clinical translation where the probe and scanners are already available for clinical use. And I'd like to conclude my presentation by suggesting where we can go from here for the reporter gene-based imaging of therapeutic immune cells. We can build a synthetic circuit to improve or do more things. For example, the circuit that can respond to activation status of therapeutic cells, so we can not only quantify and localize our infused cell, but also determine the status of their functionality. We can build multifunctional static reporters that enables imaging, long-term maintenance, and control of infiltration, etc. We can build in safety features, for example, tumor-specific promoter-driven suicide gene, or build the genetic circuit to limit cytokine production on non-lethal level. Current method to introduce our genetic material is viral-based, and there are many efforts to use non-viral transfection when we engineer this therapeutic immune cells to minimize unwanted side effects. I'd like to acknowledge all the personnel who participated in our PSMA CAR-T study. And thank you for watching our webinar series.